Hello, everyone. It looks like I am live. It is another Thursday. We are back with another biotechnology live stream. For those of you who have joined us before, I think you know me, but for anyone who is new, my name is Dr. Danielle Snowflack, and I am the Senior Director of Education at Edvotek. And I'm just really happy to be here with you today, talking about DNA, which is a favorite topic of mine. We'll talk some history, we'll talk some chemistry, we'll have a lot of fun with DNA extraction, and we'll look at some models. And we'll also have a couple fun free activities that you can do for DNA Day in your classroom, which is coming up on Monday. And I know we always like to have some fun activities, and you cannot beat free, right? Or minimal cost, but that one is delicious. So without further ado, let's get started. For those of you who may be joining us for the first time, we are Edvotek. We are the biotechnology education company. And we were founded over 30 years ago now by Dr. Jack Trichian, who was a professor of biochemistry at Georgetown University. And for him, he was in the research lab using all these fantastic technologies that were being developed and wondering why none of this was being translated into the teaching classroom, where we could really capture the imagination of students and educate a new segment of the workforce in these amazing new biotechnology techniques. And if you think about it, there were so many fantastic things happening at the time. DNA fingerprinting was becoming common, the development of the polymerase chain reaction, or PCR, which then won the Nobel Prize. That is technology we can bring into our classroom. But at the time, little of it was being translated for students. And thus, Edvetec was born. And so our focus, our main focus is on the education sphere, but we work with outreach spaces, we work with biohackers, we work with research labs, basically anyone who wants to do high quality biotechnology experiments, at a price point that's more approachable, but also for people who want to work with students to demystify science and to foster the next generation of scientists through hands-on active learning activities. So let's talk DNA. That's what we're here to talk about today, right? DNA is a favorite molecule of mine, and it's an essential molecule for all of us. And so if we think about it, the basic unit of all living organisms, from bacteria to humans, is the cell. And contained within the cell is a molecule called deoxyribonucleic acid, or DNA. Today, we know that DNA is the blueprint used to build an organism. So the directions coded for within our genetic makeup, or our genotype, is going to control our phenotype, or the observable characteristics of an organism. The directions coded for in our DNA by our genes controls everything from growth and development to cell specification, neuronal function, metabolism, our immune response to germs, and pretty much anything you can think of. And so DNA is a pretty important molecule, to say the least. And so today we are focusing again on DNA because there is a special scientific holiday coming up, which is a favorite of mine every year. It is DNA Day. And DNA Day is on April 25th every year, and it is going to celebrate two pivotal moments in DNA history. The first being the publishing of the Nature paper that described the DNA double helix in 1953. And second, the completion of the draft Human Genome Project in 2003. And that was a big day for me because I was actually in college at that point, dating myself here. But I was in college, I was a senior at that point when it came out, and I was in a genetics class. And so it was a really big moment and a really exciting moment in my own personal journey as a scientist. And I'll tell you a little bit of the ways that it can be a pivotal moment in your students' science identity as well. And so in today's experiment, we are going to extract some long polymers of DNA from solution. Historically, purified DNA was used to determine the structure and function of DNA and to learn a lot about the molecule as we were characterizing it, as scientists were characterizing it. And then we can take that DNA, and that DNA can be sequenced. And that is what happened with the Human Genome Project, where we wanted to identify each base within the human blueprint. And so today, and you may be doing this in your class already, we can use purified DNA in a lot of different contexts, including genetic engineering, transformation, restriction digest, PCR, and more. And a lot of these techniques, like I said, you're probably already doing them in your classroom, or you're looking for ways to bring them into your classroom, and hopefully you'll learn at least one today. And so, coupled with this hands-on experiment, I am going to highlight a bunch of free resources that can be coupled with your DNA experiments to celebrate DNA Day. This demonstration is being recorded, and the slides will be available on our website. I can also send them to you. Maria did put the link in the chat. So that's going to be to a form that will allow you to get the slides. I'll email them to you. And for anyone who attends live, I will also send a professional development certificate if you'd like one. That link is only going to be alive for an hour, though, after the presentation. So be sure to complete it before 4 p.m. Eastern Time. So let's talk about the history of DNA, our wonderful DNA molecule. Oh, this is the DNA extraction that we're going to be doing. And you can see that white DNA spiraled on the end of the stick there. Sometimes I forget I put animations in my presentations. So let's talk about the discovery of DNA, this wonderful molecule that we're celebrating today. The Swiss physician Friedrich Michler discovered DNA first in 1868. Cell biologists at this time wanted to learn more about organelles, and Michler was interested in learning more about the nucleus of the cell and what its function was. Remember, this was a time when we're really starting to be able to look at cells microscopically and to start to deduce the function of some of these organelles. The nucleus itself is a large, round, in some cases, dense organelle surrounded by membranes. But what was inside? And so Michler turned to white blood cells to purify nuclei. And because if you look at the slide here, you can see that those white blood cells have large nuclei that are going to be easy to purify and to get. And so Michler turned to a sort of gross starting material, but it was one that was very abundant at the time. And that was pus. So as we know, when you have an infection, your body, as we know today, when you have an infection, your body is going to send white blood cells to the source of that infection to, be able to attack and kill and neutralize any, pathogens that may be in that area. And we know that white blood cells like neutrophils are going to represent a large part of the makeup of, pus. And so Michler went to his local hospital to get used bandages. So this was a biological material that was abundant, and he was able to extract many nuclei. He carefully washed the cells from the bandages using a series of salt solutions and then burst or lysed the cells to separate the nuclei from the cytoplasm and the other organelles that were within the cell. He used a series of steps which included alkaline extraction and solution acidification, so changes in pH between acid and base to control how the DNA comes out of solution, and that resulted in the precipitation of a novel substance. This molecule, which he called nuclein, had chemical properties unlike any other substance previously identified. By the end of the 19th century, scientists had described DNA as a polymer of building blocks known as nucleotides. Many scientists, however, they had identified what nucleotides were and what they were, but most scientists believe that DNA was too simple to comprise the genetic material in a cell. Proteins at this time were also discovered, and their sequence was much more complicated. Their structure was much more complicated, and so scientists believed that this must be the material that was responsible for building an organism. And so the biological importance of DNA was not realized until much later. In an experiment that you've probably taught many times to your students. And this is going to be the experiments of Frederick Griffith. And he was trying to learn more about the epidemiology and pathology of bacterial pneumonia, which is called by Streptococcus pneumoniae. So there are several strains of this bacteria, some of which are harmless or non-pathogenic. So if they're in your body, they're not going to make you sick. And others which can cause disease, and serious disease at that. And so looking at the disease from an epidemiological perspective, Griffin found pathogenic and non-pathogenic bacteria present in patient samples over the course of a case of pneumonia. He hypothesized that one strain of pneumococcus may change into another rather than simply having multiple types of bacteria present all along. And so, following the scientific process, we have a hypothesis, we need to test it. And so in his research, he observed that living cultures of a normally non-pathogenic strain of bacteria were able to kill mice, but only after being mixed with the heat-killed strain, pathogenic strain. And so the heat-killed strain was non-pathogenic. You could inject it into mice and they would live. But when we mixed a rough strain, a non-pathogenic strain that was live with a heat-killed smooth strain, what Griffith found was that he was able to kill mice. Hi, thank you so much for joining us from Jamaica. That's amazing. This is one of the reasons why I love doing these online workshops, is because we can get people joining in and learning about DNA from all over the world. So thank you. If you have any questions, don't hesitate to stop me. I love to talk about science. And so basically, after performing these experiments, and because the non-pathogenic strain had been changed or transformed into a pathogenic strain of bacteria, Griffin named this transfer of virulence transformation. And so researchers postulated that this occurred through the transfer of genetic material, whatever the genetic material was. You have to remember, at this time, a lot of researchers thought it was protein. And so again, the protein was abundant within cells. The amino acid sequences of the proteins were very diverse. And in contrast, DNA was a much simpler molecule. There were only 4 bases linked together. And so in 1944, Oswald Avery and his group took this study one step further. So based off of the results of the Griffin study, we moved one step further. And to determine which biomolecule was responsible for transformation and which molecule was the genetic material. So Avery and his group took purified DNA, RNA, and protein from the virulent strain of S. pneumonia to determine which was responsible for transformation. The purified biomolecules were separately added to cultures of bacteria, and then those cultures were used to infect mice. And so only the recipient cells, the non-pathogenic recipient cells who were treated with the DNA molecule, were able to become pathogenic and kill the mice, leading to the recognition of DNA as the genetic material. And so that's a pretty simple experiment that allowed us to go from isolating the DNA and isolating these components to determining which was able to actually confer a genetic difference. And in another classic set of experiments, Alfred Hershey and his assistant Martha Chase used bacteria and bacteria-infective viruses called bacteriophages to reinforce the findings that DNA was a genetic material contained within cells. Bacteriophages are pretty simple viruses. In most cases, they have viral capsid proteins which surround and protect the DNA. And in one of these viruses, we were able to label the capsid proteins with a specific marker. In a second virus, the DNA was labeled. So if you can see here, we have a radio marker, a sulfur-labeled protein on the red bacteriophage, and then we have the DNA labeled with a radioactive phosphorus, and that's in green. Basically, if we mix the viruses with bacteria, which of these molecules would be incorporated into the bacteria and which would transform that bacteria and infect it? And so what they found was in the viruses that were labeled where the protein was labeled, that protein was not found, that protein was not found in the cell. But when we infect the bacteria with a virus with the labeled DNA, that DNA was found into the cell, and that was able to infect the cell. And so this experiment basically showed us that DNA was the genetic material, and that was being passed into bacteria, and that was able to essentially reprogram the bacteria. Another important set of another important set of observations were made by Erwin Chargaff in the late 1940s. So let's step it back again. Scientists realized that DNA was made of nucleotides, our adenine, thymine, cytosine, guanine, there are 4 nucleotides. Again, and they were starting to learn that this was the genetic material. So how can we go from this simple genetic material to a genetic code? And so one of the things that Chargaff noticed in his research was that normal DNA, isolated from any species of organism, and this is from bacteria to human, had a 1-to-1 ratio of purine bases to pyrimidine bases. So purines are our 2-ring bases, our adenine and our guanosine. Pyrimidines are our single rings, so our thymine and our cytosine. And so we were able to find these in a specific ratio. Again, it was a 1-to-1. So for every pyrimidine, we had a purine. And no one really understood why this was, but it was observed across all species. And so this observation and the others that we had talked about, plus one more, would prove important to solving one of the great mysteries, one of the great questions in biology at that time. What is the structure of DNA and how does that structure tell us what's going on? And so these experiments kicked off a worldwide race to unlock the secrets of our DNA, starting with its structure. Hello, another person. Thank you so much for joining us. I always love it when people say hi to me in chat. Welcome also, to today's DNA Day celebration. And I hope that, you learn something and find some experiments that will be able to work in your classroom. So as I mentioned, the experiments that the experiments that we just discussed really helped to kick off a worldwide race to unlock the secrets of what's going on in our DNA, starting with its structure. And thus we arrive at the story of James Watson and Francis Crick, who first described the double helix of DNA in a 1953 Nature paper. And so that's the April 25th, 1953 date, which helped to inform the April 25th date of DNA Day. And so one common misconception that I do think is being addressed today in science classrooms, but this common misconception, and it's one that I had, in my formative scientific experiences, was that Watson and Crick were the people who discovered DNA, which we know isn't true. Instead, what they were able to do was to collect all of the data that people had learned about DNA, from its function to its structure to observations of its chemical properties, and use that to create the simple and elegant model of the DNA double helix, which we know today. And I have a model here of DNA. I can switch over to the model so that we can look at the DNA structure. So let's bring this forward a little bit and we can focus on it. So here's our DNA structure that we know today. And there were a lot of really important pieces of data that came together to allow us to determine the double helical structure of DNA and the way the bases come together. Watson and Crick were the first to describe the double helix, and I think this is a really important work and worthy of the Nobel Prize. But I do always think it's important to mention that this work was dependent on the work of those who came before, and they weren't always rightly credited with the work that they did. The picture that we see here, this is the famous photo 51. And this was an X-ray diffraction image taken by Rosalind Franklin and her student Raymond Gosling. And so this photo, based on what is seen in it, was one piece of data that came together with work from Wilkins, Stokes, and Wilson, who were also modeling DNA structure at that time in the same location. And then additionally, because of Chargaff's rules, which I meant to spell out a little bit further here, but I just put Chargaff rules and I misspelled it as well. So I will fix that when I send out this slide deck. Again, if you want the slides, please be sure to fill out the form and I will send them to you. I will fix that. But again, because of Chargaff's rules, when we take those rules and look at this picture and bring together the data, it makes sense because we can then elucidate that hydrogen bonds between nucleotides hold two strands of DNA together. And one pyrimidine is always going to match with one purine. So A to T and C to G. And we always have a purine with a pyrimidine, and that's going to give us a single unit or a rung in our ladder. And because it's always a molecule with one ring and then two rings, the width of our DNA double helix is going to be the same, from base to base. And let me actually take my model here and we'll look at it really quickly. We can see each of these is a DNA base pair, blue and red, and green and yellow represent different bases. And if we look here from the top down, what we see is that we have and again, this is obviously a model, but we can see we have a constant width. Each nucleotide pair is a constant width. And that gives us a really nice ordered structure. Based on the way the molecules come together, we have a major groove and a minor groove, and these are important for the way that proteins interact with the DNA and allow it to be read and processed. So now we know that DNA has a specific structure and it's built from nucleotides. Chemically, each nucleotide has three basic parts. We have a phosphate group, a deoxyribose sugar, and a nitrogenous base. Those are our A's and our T's, our C's and our G's. And those all come together to create a nucleotide. The sugar of one nucleotide is going to be bonded to the phosphate of its neighbor. And that is going to be the way the molecules bind. And based on the chemical properties, that does give us a directionality to each strand of DNA. And that's important for the way that it's read. It's always read from the 5' end to the 3' end by the molecules, the enzymes in our body that read DNA. But because of this backbone, and we know we have the bases come together, they're held together by hydrogen bonds through the side chains in the chemical molecules, but also the chemistry of the backbone is important itself too. Because of the nature of the chemistry, we have a strong negative charge, which is going to make it very soluble in water. And that's a good thing because humans, our bodies are largely made of water and our cells are largely made of water. And we want to make sure the DNA that's in our body is also soluble in water. Hello. Thank you. It's always so nice to have more people join the workshop. If you guys have any questions, if you people have any questions at any time, please let me know. I am happy to answer them. So again, the DNA genome is an organism's instruction book. The blueprint contained within each of our cells, the instructions in each of our cell code for structural proteins and necessary enzyme. Some DNA is responsible for genes, which is a discrete sequence of nucleotides. The DNA is transcribed into RNA and then translated into protein. So basically, a gene sequence is a recipe that tells us how to build a protein. Oh, thank you. Yes, I do think it's really important that Rosalind Franklin and others are really emphasized in scientific discovery. I do always put the picture of Hershey and Chase up too, because it's another place where, a female was involved in a pivotal scientific description. And I always do try to highlight, people who are, underrepresented or unknown in all biology classrooms. And so I always like to highlight everybody who is involved in the process and try to highlight more and learn more myself. So in bacterial cells, I'm getting a little bit of buffering. Are you all able to see me and hear me okay? Definitely let me know if you're having problems. I'm going to keep going though. So definitely, if you're having problems, let me know in chat and I will try and fix some settings. In bacterial cells, we find that long threads of DNA are organized into circular chromosomes. We also find that plasmids are within bacteria. A plasmid is a small circular piece of double-stranded DNA found in addition to their chromosomal DNA. Plasmids can contain extra non-essential genes which are passed between bacteria, and an example of this is antibiotic resistance. In eukaryotic cells, we find the majority of cellular DNA in the nucleus as chromosomes, though mitochondria and chloroplasts have their own mini genomes, which I think is completely cool. Some eukaryotic cells can contain plasmids as well. And for example, plasmids are commonly used for genetically engineering yeast cells. Furthermore, some viruses use DNA as the genetic material, but not exclusively. And viruses are these simple infectious particles that replicate in a host cell. Some use DNA as their genetic material, others use RNA. In general, they're not considered to be living because they need host cells to multiply. So before we perform many important molecular biology and biotechnology experiments, including sequencing our DNA, we have to isolate the DNA from cells. And like the researchers before us, we'll use the chemical properties of DNA to separate it from the other components of cells. And so what do we need to extract DNA from cells? So let me show you what we have in this experiment. So first things first, and you may already be extracting DNA. You may be using a different experiment, or you have a favorite one that you do at home with alcohol and Dawn dish detergent. That's very common. The kit we're doing today is one where the DNA solution it's going to be this one, Edvotek kit S10. What does DNA look like? This is one that includes the DNA as a concentrate that's already been extracted. Can you see that? It includes a buffer. And so the buffer, the DNA extraction buffer in general, is something that is going to allow us to lyse cells or break them open. It breaks down cell and nuclear membranes, allowing the DNA to be released. And most buffers that are used in DNA extraction, can we see this here, are going to have a couple key components. We have a buffer to stabilize the sample's pH, a chelating agent to activate DNases, which are enzymes that like to chew up and break down DNA, a detergent to dissolve lipids like the cell membrane, and that's going to be like your Dawn dish detergent, and then salt, which is going to disrupt the interactions between DNA and water. We have various kinds of tubes. I'm going to be using cups today. These little medicine cups come with the kit. Where's my camera? There's my camera. And these are going to be our vessel to perform the extraction. We have spooling rods. These are great. This is a glass spooling rod. You can also use wooden coffee stirrers, plastic coffee stirrers, really whatever you have on hand that's kind of rough and a little sticky. It gets the DNA stuck on there. We have ice cold isopropyl alcohol. Here is my little live stream ice bucket that I have here. Let's not do any advertising for anybody. I'm doing this from my basement, from my home, so we're limited. So this happens to be the ice bucket that I have. But I know you're all teachers or educators, and so you are also plucky and use what you have on hand. Then we have our DNA solution here, which I've prepared, again, in a jar that I had at home. And then we need our calibrated transfer pipettes, which I have here. Pipettes are just a great way for us to be able to move liquids from tube to tube. These in particular are calibrated with lines on the side which allow us to make measurements. And so to extract DNA from our cells, we need to break open the cell wall and membranes, the nuclear membrane, and then separate the DNA from all the other gunk, the organelles, proteins that are present in the cytoplasm. And we do this in a process called lysis. So if you are in your classroom and you are doing strawberry DNA extraction, which is common experiment, you would then basically take your plant sample, crush it in the DNA buffer, and the combination of the salt, the detergent, and the physical crushing of the cells is going to liberate DNA from the cells. So we talked about extraction buffer in the last slide. Where did I put that? We talked about our buffer in the last slide. This came as a concentrate that you just dilute in water. So it's really easy, and that allows us to, get you the experiment in a small box instead of a big box, which helps with shipping. So we provide everything as a concentrate. As a reminder, an extraction buffer, we have salt, which salt is, in this case, sodium chloride is a major component of the buffer. In a solution of sodium chloride, the sodium chloride molecules separate into sodium ions and chloride ions. Sodium is a positively charged ion. DNA, on the other hand, is highly negative charged. And again, that is because of the sugar phosphate backbone. This large negative charge of the DNA molecule is neutralized by the positive sodium ions in solution. This disrupts the interactions between DNA molecules and water, making DNA less soluble in the water. Without the salt, the DNA remains negatively charged and it's happy in our aqueous solution. Next, we add ice-cold isopropyl alcohol to our cell lysate. DNA is not very soluble in isopropanol, especially when the negatively charged backbone is neutralized by salt. So this causes the DNA to precipitate or kind of crash out of the solution of the cellular lysate as sticky white fibers. Furthermore, we make sure our alcohol is super cold. Again, here's my makeshift ice bucket, and you can see I have this good crushed ice that I've been keeping my alcohol cold in. It might not be perfect because it has been melting a little bit under this light, but it is cold, cold, cold. DNA is even less soluble. Is even less soluble in the cold alcohol, and so that helps us promote precipitation. If the mixture is mixed and centrifuged, the precipitated DNA will become a pellet at the bottom of the tube. In this experiment, we're going to carefully layer our isopropyl alcohol on top of the DNA solution where it precipitates at the interface, and then we use a spooling rod to pull the DNA out of the tube. So let's do it. Okay, so we collect our reagents. You always want to make sure to have your reagents on hand. You're always going to want to use proper personal protective equipment. Nothing I'm using is particularly hazardous today. I always like to wear gloves. It's always important to protect you from the science and the science from you. We're trying to keep our DNases down, but we have cells on our fingers. The cells, we know skin cells are always sloughing off. There's a possibility for there to be DNases in those cells. That's going to ruin your experiment. So always use good personal protective equipment, gloves, goggles, lab coats, whatever is suitable for the experiment. So we collect our reagents. I've got my DNA solution here. I've got my alcohol. I've got my pipette. And so what I'm going to do is let me aim this camera down a little bit. All right, so we can see the cup. All right, here's the cup. You should all be able to see that. So I'm going to start by adding some DNA from my jar to here. And now if you're in your classroom, you'd be measuring a lot more carefully. You'd have this all pre-aliquoted for your students. I am being the bad teacher today. So I am just pouring it out by hand. I'm going to take my cold alcohol. And we're going to use the pipette. And we want to carefully layer this alcohol on top of our DNA. And we're going to do that by just gently turning this on its side and slowly and gently adding the alcohol. And the important thing is to make sure that you have an interface. So that is probably, oh no, we can see it. So you should be able to see very slightly right about here. There is an interface where, we have a different optical density of these two substances. And that is where our precipitation is happening. Let me get a little bit more alcohol, which is very cold from the bottom. I'm going to layer just a little bit more on top. And then here we go. The magic of DNA. So I'm going to take the end of the spooling rod below the interface where the two come together, and we start spinning and we start twirling. And what we can start to see is that we get goopy threads. Oh, can you see that goopiness? Oh no, am I off camera? Can you see that goopiness? That is our DNA. Let me get more. There we go. Do you see that goopiness hanging off? That is going to be the DNA. And it's a little subtle when you're doing it on camera because we're looking at a clear, colorless substance that is, coming out of a clear, colorless substance. But what I hope you can start to see is that we're getting this goopiness, and that's our DNA. So DNA is a polymer. A polymer is basically a string of nucleotides that are stuck to one another. And if you look carefully, you can see at the end of this spooling rod that there is a thickness. And that is our DNA. Let me see where, if I put it right here, you can start to see that thickness at the end of the string. And that's our DNA. Let me see if I can get even more out of here. Yeah, you can see that stringy goopiness. And so I personally could continue spooling DNA all day. I think it's just so cool. I'm the only scientist in my family. And I remember one time my brother did this experiment in high school or in college. And he said, Danny, I didn't know that DNA looked like snot, like boogers. And it really does. It is a clear, sticky, goopy substance that comes out of the solution. And amazes me every time I do this experiment. So let me see if I could get a really good goopy. Okay, so again, let me see if I put something blue behind it. If we look here, we can see that goopy whiteness. And that is our DNA. Cool. I always love to look at DNA. There are lots of, let's see. So now that we have the DNA on our stick. If we were doing biotechnology applications, I just knocked my light off. If we were doing biotechnology applications, we would generally extract it we would perform this in a centrifuge tube, spin it down, and get a purified sample of DNA. And so what do we want to do with that DNA? And this is where we start to talk about, DNA sequencing and learning more about the human genome, which is the second part of our fantastic DNA Day celebration. Why can't I get this to stay here? Oh well. If you want to see it again, I can spool again too. But we have a bunch of some other stuff that I do want to get to. So we want to learn about the human genome. What we need to do is have a strategy that we can learn each letter. And that is the way that we sequence DNA. And the way I'm going to talk about is Sanger DNA sequencing. This technique allows us to determine the identity of every nucleotide in a DNA sequence. We target genes using a short synthetic primer that binds somewhere within a specific DNA sequence. The sample to be sequenced is then combined with a primer, the DNA building enzyme DNA polymerase 1, and a blend of nucleotides. So those are our building blocks, our A's, our G's, our C's, and our T's. The mixture includes a high concentration of DNTPs or deoxynucleotides and a low concentration of a nucleotide analog called a DDNTP or dideoxynucleotide. And so these DDNTPs are nucleotide analogs that lack the 3' hydroxyl group. And it's important to note that this group is missing. And so we can see it when we look at the two figures that are here. When we get rid of that 3' hydroxyl group, DNA polymerase cannot attach another nucleotide to the growing DNA strand. So if we have this here, if we look at our DNA structure again, right? So we have our DNA structure and you can see that our sugar is connected to our phosphate, is connected to our sugar, is connected to our phosphate, and all the way through linking the DNA. If we were to have a nucleotide that was not able to make that linkage, we would not be able to extend our piece of DNA. And that's important when we're talking about Sanger DNA sequencing. Because during our sequencing reaction, DNA polymerase 1 is going to read the DNA. And so a lot of the times we talk about, the initial sequencing experiments done in the 60s, 70s, 80s were done with gels. Today, we actually can use fluorescently labeled nucleotides. And those allow us to create chromatograms that tell us the sequence in a lot easier fashion than having to look at a DNA sequence and read the letters and interpret it that way. So during our sequencing reaction, DNA polymerase 1 reads the DNA template and adds nucleotides to the primer. Can everybody see okay? Is there a problem? So if we're building DNA, be sure to let me know in the chat if there are problems. I'm not buffering right now, so it should be okay. So during the sequence reaction, DNA polymerase 1 is going to read the DNA template and add nucleotides to the primer to build a complementary strand of DNA. And so as I mentioned on our last slide real quick, we make these sequencing reactions. Oop, that's my face. I wanted to go back to the last slide. We have these sequencing reactions that contain free nucleotides and that also have these special dideoxynucleotides. And so we have a high concentration of DNTPs and a very low concentration of DDNTPs. So most of the time when polymerase is building DNA, it will just add the next nucleotide to the growing piece of DNA. However, when DNA polymerase 1 adds a DDNTP to the DNA strand, it's impossible for DNA polymerase to add another nucleotide to the end of the growing strand. And again, this is because we're missing that 3' hydroxyl group that is used to link our two nucleotides together. This is going to stop the growth of our DNA chain, and that creates a series of fragments of different sizes. Ideally, we get a stop after each base, and that is how we sequence DNA. The site of the dideoxynucleotide incorporation allows one to determine the position of that base. The shortest fragments result from DNA strands that added the nucleotide near the primer, where the longer fragments had more DNTPs linked to the growing DNA chain before the incorporation of the DDNTP. To read the sequence, the DNA fragments are separated by capillary electrophoresis. Automated machines separate DNA fragments through a polyacrylamide gel formed in a thin capillary tube. Importantly, each DDNTP is labeled with a different fluorescent marker, allowing the sequence of a particular DNA strand to be read by a laser that's focused on the capillary. As the DNA fragments pass through the gel matrix, the laser excites the fluorophores on the DNA. The 4 different DDNTP fluorescence colors are then automatically detected, and the fluorescence intensity is translated to a data peak. And so this became really important to determine the sequence of DNA. So the Human Genome Project was begun in the early 90s, and it concluded with the sequence of the DNA of the human genome. And this was an international endeavor to characterize what's in our cells, what is in there that tells our body, what directions do we have in our cells that tell us how to build an organism, and how to tell the organism that we are, the animal that we are, to continue to grow and to thrive and to live. And so there were the initial approach taken by the Human Genome Project was called contig sequencing or genome walking. And basically what would happen in this project was that researchers took human DNA from many donors, and they cut it into large pieces. Those fragments are cloned into bacterial artificial chromosomes, or BACs. And these are basically like plasmids, like small pieces, like pieces of DNA that are in bacteria that can be replicated inside of the bacteria, but they can hold really large sequences of DNA. And they call them artificial chromosomes because they're circular and they're similar to a bacterial chromosome. And we know what DNA is in the back. We have some landmarks in that DNA that we can use. And then what we do is we cut the sequence within the back into smaller pieces and use Sanger sequencing to sequence them. The sequences are then assembled using computers. And at the time in the 90s when this was being done, the computers were taking up entire rooms to be able to process the amount of data that they needed to be able to process. Today, we'll talk about today in a minute, but and these computers would then be used to read the sequences, find out where the overlaps are, and put them together, allowing us to compile a sequence. Since all the pieces in a single back are close to each other, they're from the same region of the genome, it's easy to assemble the DNA sequence from these overlapping pieces of DNA. There are also markers that can be used as landmarks for the assembly. But there is a disadvantage in that these processing steps can take a long time. And so to get a good usable sequence from this small region of DNA, it takes a lot of time. And this was the approach taken by Francis Collins, a name you may know from his time as the director of the NIH and also now his role in the current administration, with science policy. And so, he got to start. He got to start many years before the Human Genome Program, but I think he became a household name with his work on this project. But there was a second person, J. Craig Venter, who was a biotech entrepreneur who wanted to take a different approach to genome sequencing. And he and his group did what's called shotgun sequencing. And so basically you're starting with the same genomic DNA, the same material from humans, and this was sheared into very small pieces that were then sequenced. So basically we're skipping out on all the intermediate steps of the backs, and the chromosome walking. And then the pieces, the sequences are assembled. And so you can imagine that this is very fast because we're getting rid of a lot of the interpretation steps in the middle. But sequence assembly is way more complicated. We can't walk the chromosome. We don't know the region where these sequences are coming from to be able to create the final human genome. So now I'm buffering. So if anybody's having problems seeing me, I am seeing that I am buffering a little bit. Okay, I'm back. So just to tie up some loose ends, the human genome was begun in the early 1990s and quote unquote completed in 2003. And this is what I was saying, where it was a momentous occasion for me, as a person studying genetics, because this was the sequence of the human genome. And that was a momentous occasion. And it came in early and under budget, which never happens with projects. However, in reality, that was only 85% of the sequence. And so we know that there are repetitive regions of DNA. We know that DNA is very condensed around the centromeres. We know that DNA sequence is repetitive at the telomeres. And so really in 2003, when I thought the genome was published, it was only about 85% of the sequence. And so recently, in the past few years, the more difficult regions of the DNA, including centromeric DNA, telomeric DNA, and the sex chromosomes, were able to be fully sequenced. And one example, the Y chromosome was probably the most difficult chromosome to sequence because it has a lot of repetitive DNA and heterochromatic DNA, which made it really difficult to figure out the order of the DNA nucleotides. But recently, that changed. And so papers were published showing what they called T to T DNA sequence, or telomere to telomere DNA sequence, end-to-end sequence. And so that was actually just published very recently. And the two articles here are references that you can give to your students to tell them about, what is still going on in the lab. Like, I always think it's amazing that things we kind of think are canon, our knowledge, we're still learning, and scientists are still developing theories and models for what they are. And so again, if you get these slides, you can definitely download from here. Oh, yeah, so this is a funny tweet that I saw that I thought was, am I stuck? Can you see me? Huh. Oh, no, you can see me. Okay. I had a message that I was stuck, but I am not stuck anymore. And so this was a funny meme that I saw. It was a tweet from NHGRI, which is the directorate within NIH that was responsible for the Human Genome Project, where they were talking about the T2T consortium. And, I always like to use memes when I'm teaching because I think that students relate to it. And I think teachers relate to good memes too. So let's talk about, the DNA extraction experiment we did came with a concentrated DNA solution. We have lots of options at Edvotek if you want to extract DNA. Here are just a few of them. One experiment that's might be a little too hard to do on Monday, but next year for DNA Day, keep in mind, it's exploring the genetics of taste, where we look at the PTC gene, we figure out our genotype using PCR, and we extract the DNA, and it's just a really fun one to link genotype and phenotype. And now to the free resources, which I'm going to show you. There's, of course, and this is completely clickable. So when I send you the slides, you'll be able to link out to all these resources. If Maria wants to, maybe she can link to our candy molecule protocol, our word search, the DNA structure worksheet, or at least just the guides and lessons plan page so that people can take a look at that. But basically, one of the best resources for learning about DNA Day is to go to the official DNA Day website at NHGRI. But on our website, we do have a protocol for one of my favorite experiments that I'll run through really quickly, which is creating a DNA model from candy. And so this is a fun experiment to do with your students. We have this really nice printable page available for free on our website that give directions for how to build your own DNA from candy. And so I have some toothpicks and I have some dots, and we're going to just build some candy really quickly while we have a few minutes left together. So I have dots. This was, believe it or not, I could not find gumdrops at my local drugstore. So this is the gummy candy that I have. And we have 5 colors. So let me see what colors I can find. All right. So of course, you're generally not going to want to do this on the same bench with your other chemical reagents, but I'm a little bit limited for space. So let's build our DNA. I have dots for DNA for our nucleotides. And then I have Twizzlers here for our DNA backbone, our sugar phosphate bonds. And so this is just an easy, fun snack time for your students where they can build DNA. So I'm going to pair, I'm going to pair red and green. And I'm going to pair orange and pink. It's going to be a little subtle, but for some reason I did not get any. I only got one orange yellow. So maybe if I do red and pink with each other. Let's see, no. You'll be able to select your candy a little bit more carefully than I am. Hopefully get a couple more colors. But what we're going to do is we are going to skewer our nucleotides together. So let's say we've got an A and a T here. And we're just making these little skewers. And because we're using toothpicks, the distance between our nucleotide backbones is going to be consistent from sample to sample. Let's have a green and a red. And we're just going to skewer these on again. And this is fun if you have the opportunity to have a little party because, you know, your students can build some DNA and then have a little treat. I have seen adults do outreach events where they do DNA extraction using strawberries and use it to make a strawberry daiquiri cocktail. You're probably not going to do that with the students because you do not want to introduce them to ethanol in your classroom in the way that a daiquiri would. But I know a lot of people who are doing outreach consider that a fun activity to do. You'd have to look online for a protocol to extract DNA without a ton of salt and detergent though, because we wouldn't want to drink that. But this could be a fun activity to do, after your students extract DNA, or actually before your students extract DNA, they can build their own model of DNA. And think about how would they have determined the structure. All right, so here we have some nucleotides, and I'm excited to be able to eat these candies later as well. So make sure to pick something you like. And so we're just attaching our backbone. I'm just doing 4 in the interest of time, but what you can see is now I have my DNA kind of ladder. I have my 2 sugar phosphate backbones, and I have my DNA nucleotides. And we can actually use that and twist it and start to create, a helical shape. And so I just think that's fun, a fun way for students to model DNA. We have, and this again is a free resource available on our website. It'll be linked to, if you request the slide deck, I'll send that to you, tomorrow morning, first thing. But it's also available on our website if you go to edvotek.com and look for our guides and lesson plans. So this is a free printable that you'll be able to use. We have other free printables that are fun. This one is also going to focus on the structure of DNA. It's a coloring page showing how DNA is bonded. It shows our different 5' to 3' sides, the hydrogen bonds between the molecules, and then your students can, pick a color for each of the bases, and then color it in and explore the structure of DNA that way. This is a fun activity to be done, in combination with the DNA model. We have coloring sheets with DNA electrophoresis. This is fun. If you've done electrophoresis, your students can create an experiment and show the results here, as part of their lab notebook, but they can also potentially, use this just as a fun DNA day activity. What kind of gel would you like to see? And then the last link here is a fun one from a professional society, the American Society for Biochemistry and Molecular Biology, where you can use beads and elastic to create DNA sequence bracelets. And so you could even use some of those sequences to create candy DNA as well. Basically, there are sequences like, for example, this is a short sequence from firefly DNA. That is CAATTGTA. And so the cards talk a little bit about what the gene does and shows the sequence. And you can use that to create a model. The DNA bracelet only shows one strand, so you could have your students have to think about, what base pairs together to build the second strand. So that's a fun extension of those activities. And so hopefully in this live stream, you learned a lot about DNA. And you have some resources to help you explore and be excited about it for your students for DNA Day, which is actually coming up next week on Monday, April 25th. If you have any questions now, definitely put them in the chat because I am going to start wrapping up and I want to make sure I get to anything that you might have. But DNA, candy or otherwise, is the genetic material found in living organisms and in some viruses. And it's the blueprint that's going to tell an organism how to function, how to build itself, how to maintain itself, and how to grow new organisms. Many experiments were performed to identify the DNA as the genetic material and to unlock what it means. And so that goes back to the first experiments extracting DNA from the nucleus, goes through to the fact where we figured out DNA was the genetic material, and then learning about its structure and its sequence and what that tells us about DNA and ourselves. One way we can explore DNA in the classroom is through DNA extraction. And the extraction I showed you today was a quick one using alcohol precipitation. So I used isopropyl alcohol. Let me see if I can get some more DNA out of here, just for a wrap up. Oh, there's more. And so, we can extract DNA using alcohol precipitation. Again, spooling on a stick is not the kind of extraction that you would be doing today. For more sequencing, this is pretty well exhausted, but you wouldn't be spooling DNA for sequencing applications, but it's a fun way to show that cells have DNA and what DNA looks like and how DNA is a long, sticky, goopy polymer. And DNA Day, I think, is a fun holiday for the biotechnology classroom, for the biology classroom, to really be able to be excited and to discuss some of these important milestones in DNA history and the people who were pivotal in DNA history. So with that, please be sure to request the presentation. We're here on YouTube monthly, but you can find us in a lot of different places. Our YouTube channel, please like this video and please be sure to subscribe to our channel. We're always trying to put out videos and tutorials to help our educators, get their students excited about science. We're on Facebook, we're on Twitter, we're on Instagram. I forgot to put TikTok on this slide, but we are on TikTok too. We release a new TikTok every Friday, which is totally fun. And it's certainly a media that I don't know if I understand entirely, but I know our students do. They live and breathe TikTok. And so that is fun. And so I just want to take this opportunity. If you have not signed up yet, please be sure to sign up to get the slide deck and the professional development certificate. Thank you so much. I'm glad it's sunny in Minneapolis. I'm actually not sure what it is like here in Maryland, given that it is, given that I am in my basement. But I'm glad you like the information on sequencing. And I'm here pretty regularly. If you haven't done one of our live streams before, please be sure to check out the playlist. We have many that we have covered. And if there's a topic you want to see represented in a future live stream, just put it in that form and we will be sure to try and address it in a future live stream. So thanks again. I was really happy. These are always really happy times for me. I love talking about science. I love being, with science teachers talking about science.